Okay, we are winding down the semester. We got two chapters to cover this week. We'll start with the brain and the cranial nerves. So, the surface anatomy of the brain is best defined by these folds. We have what is known as the highly folded brain, the highest, most folded brain of all the vertebrates. And those folds are going to have specific names and indicate locations of things happening in the brain. We look at it from the side, we see that those holes include grooves that go deep. They're called sulci, and the upper portions are called gyri. So the brain is composed of gray and white matter. The gray matter has the nerve cell bodies, the dendrites, the synapses. Most of that is on the cortex, the surface layer of the gray matter, and the cerebrum and the cerebellum. Deeper than that, when we have gray matter, we refer to them as nuclei in the central nervous system, where we call them ganglia in the peripheral nervous system. The white matter is composed of tracts. These are bundles of nerve fibers, the axons of an individual nerve, that are coated with myelin, and they're deep to the cortical gray matter in the brain. They connect one part of the brain to another, as well as to the spinal cord. Embryonically, the nervous system develops from ectoderm, which is the outermost tissue layer of the embryo. Neurulation occurs within the first 3 weeks. It's a very early event in the development of an embryo, so lots of things that sometimes when people don't even know they're pregnant can go wrong during this initial stage. The dorsal midline of the embryo will thicken and form what is known as a neural plate. That neural plate will sink, its edges will get thicker, and it will form a neural groove with raised neural folds on each side. Eventually, those neural folds will fuse, creating a hollow neural tube by 26 days, roughly a month. The lumen of the neural tube becomes fluid-filled space, and later is going to be the ventricles of the brain and the central canal of the spinal cord. The neural crest is a longitudinal column on each side of the neural tube formed from ectodermal cells. It's going to give rise to the two inner meninges, most of the peripheral nervous system, and the other structures of the skeletal, integumentary, and endocrine system. So by the fourth week, the neural tube exhibits three primary vesicles at its anterior end: the forebrain, midbrain, and hindbrain, also called procephalon, mesencephalon, and rhombencephalon. By the fifth week, the neural tube is going to subdivide further and form 5 secondary vesicles. So the forebrain will split into the telencephalon and the diencephalon. The midbrain doesn't divide, it remains the mesencephalon. And the hindbrain divides into 2 vesicles, the metencephalon and the myelencephalon. The meninges, those coverings that surround the brain and spinal cord, they lie between the nervous tissue and the bone. We talked about them a little bit before, but they protect the brain and provide the structural framework for its arteries and vein. If we go from the outermost to the innermost, we have the dura mater, followed by the arachnoid matter and the pia mater. The dura mater has 2 layers. The outer is the periosteal layer. It's equivalent to the periosteum of the cranial bones. The inner meningeal layer continues into the vertebral canal and forms the dural sheath around the spinal cord. Dural sinuses are spaces located where the periosteal and meningeal layers separate. They collect blood circulating through the brain. The superior sagittal sinus lies just underneath the calveoli and the median line, and the transverse sinus runs horizontally from the rear of the head toward each ear. Dura mater presses closely against the cranial bones. There is no epidural space like we had in the spinal cord. It's not directly attached to the bone except around the foramen magnum, the sella turica, the crista galli, and the sutures of the skull. Folds of the dura mater extend inward, separating some of the brain regions. There's a falx cerebri separating the 2 cerebral hemispheres, a tentorium cerebelli separating the cerebrum from the cerebellum, falx cerebelli that separates the right and left half of the cerebellum. Arachnoid matter is a transparent membrane over the brain surface. The subarachnoid space separates it from the pia mater below, and that space is filled with cerebral spinal fluid and blood vessels. Directly against the brain is the pia mater, which is a very thin membrane, not usually visible without a microscope. It follows all the contours of the brain, and it follows the arteries as they penetrate into the cerebrum. The dura mater presses closely against cranial bones. Again, there are no... Whoops, did I go backwards? I did, I'm sorry. So here we have a diagram of the meninges of the brain, along with skull bones. Meningitis is an inflammation of the meninges. It's a serious disease of infancy and childhood, especially between 3 months and 2 years of age. It can be bacterial or viral in nature, and it invades the central nervous system, generally through the nose and throat. Pia mater and arachnoid matter are most often affected. It can cause swelling of the brain, enlargement of the ventricles, and hemorrhage. Signs would include high fever, stiff neck, drowsiness, intense headache. May progress to coma and then death within hours of onset. And it's diagnosed by examining cerebral spinal fluid that we obtain from a lumbar puncture or a spinal tap. Ventricles are 4 internal fluid-filled chambers of the brain. There are 2 lateral ventricles, one on each cerebral hemisphere. A third ventricle is a narrow medial space between the corpus callosum. The fourth is a small triangular chamber between the pons and the cerebellum. The ventricles are all interconnected. There's the interventricular foramen. That's a pore that connects the lateral ventricles to the third ventricle. A cerebral aqueduct is a tube that runs through the midbrain, connecting the third ventricle to the fourth ventricle. And then the central canal connects the fourth ventricle, and that runs throughout the center of the spinal cord. That cerebral spinal fluid is a clear, colorless liquid that fills the ventricles, the canals of the central nervous system. It bathes its external surface. It's where we produce, the production of cerebral spinal fluid begins with filtration of blood plasma through capillaries of the brain. The choroid plexus is a spongy mass of blood capillaries on the floor of each ventricle. The epididymal cells are the neuroglia that cover the choroid plexus. They modify the filtrate. It's very similar to plasma, but the cerebral spinal fluid has more sodium and chloride, less potassium, calcium, glucose, and very little protein. That cerebral spinal fluid flows continuously through the central nervous system. It's driven by its own pressure, also by the beating of epididymal cilia and the pulsation of the brain that's produced with heartbeats. It's secreted through the lateral ventricles, through the intervertebral foramen into the third ventricle, down the cerebral aqueduct into the fourth, and the third and fourth ventricles add more cerebral spinal fluid along the way. All of that cerebral spinal fluid ultimately escapes through 3 pores that lead into subarachnoid space of the brain and spinal cord, the median aperture and the 2 lateral apertures. It is then reabsorbed by the arachnoid granulations. Those are cauliflower-shaped extensions of the arachnoid meninx. They protrude through the dura mater into the superior sagittal sinus, and that cerebral spinal fluid penetrates the walls of the villi and mixes with the blood in the sinus. That cerebral spinal fluid provides buoyancy, protection, and chemical stability. The brain kind of floats in it, protects the brain from striking the bones as we move around, and it provides for a much more stable chemical environment than even is found within plasma. The brain only contributes about 2% of the adult body weight, but it gets 15% of the blood, approximately 750 milliliters per minute. Neurons have a high demand for ATP, and because of that, they need oxygen and glucose to supply that ATP. If we get a 10-second interruption, we can get loss of consciousness. 1 to 2 minute interruption, we can get a significant impairment of neural function. And after about 3 to 4 minutes, we get irreversible brain damage. There is a blood-brain barrier which regulates the substance that can get from the bloodstream into the tissue of the brain, the fluid of the brain. Its blood is crucial, but it also contains sometimes harmful agents. That barrier system is selectively permeable. It is very permeable to water, glucose, lipid-soluble substances like oxygen, carbon dioxide, alcohol, caffeine, nicotine, and anesthetics. It is slightly permeable to sodium, potassium, chloride, and waste products such as urea and creatinine. The points of entry must be guarded. The blood capillaries through the brain tissue guard by the blood-brain barrier, while the capillaries of the choroid plexus are guarded by the blood cerebral spinal fluid barrier. That protects the brain and its capillaries. Consists of tight junctions between the endothelial cells that form the capillary walls. Anything leaving the blood must pass through the cells and not the gaps between them. And those endothelial cells can be quite selective, unlike the gaps. You have to internalize it on one side of the cell and pass it through to the other side of the cell. It's formed by tight junctions between the epidermal cells, and tight junctions are absent from epidermal cells elsewhere. This is for the blood central cerebral spinal fluid barrier. And it's important to allow exchange between brain tissues and cerebral spinal fluid. The brain barrier system can be an obstacle for delivering medications, antibiotics, cancer drugs. Trauma and inflammation can damage the blood barrier system, allow pathogens to enter the brain tissue. There are circumventricular organs where the barrier is absent, and those would be in portions of the third and fourth ventricles. This allows blood direct access to the brain. It also enables the brain to monitor and respond to fluctuations in blood glucose, pH, osmolarity, and some other variables. Unfortunately, the CVO also provides an invasion route for human immunodeficiency virus, HIV. A stroke is a cerebral vascular accident, or CVA. It's a sudden death of brain tissue due to interruption of blood supply. It can occur at any age, but 2/3 are people 65 and older. It is the second greatest cause of death after coronary artery disease. About 50% of stroke patients live less than a year or longer. There are two possible causes for the stroke. A hemorrhagic stroke would be a ruptured vessel, while an ischemic stroke would be an obstruction of a vessel that prevents oxygen from getting to the cells. The medulla oblongata is the adult brain region that develops from the myencephalon, begins at the foramen magnum of the skull, extends about 3 centimeters rostally, and ends at a groove just below the pons. It is a little bit wider than the spinal cord. It has pyramids, which are ridges on the anterior surface that resemble side-by-side baseball bats. There are 4 pairs of cranial nerves that begin or end in the medulla, 8, 9, 10, and 12. There are structures called olids, which are prominent bulges lateral to each pyramid. The gracillae and the cuneate fascicula of the spinal cord continue as 2 pairs of ridges on posterior medulla. They contain sensory fibers and synapse in the gracilis and the cuneate nuclei. All the ascending and descending fibers connecting the brain and spinal cord pass through the medulla, like the medial lemniscus and the corticospinal tracts. The medulla contains numerous nuclei, including the inferior olivary nucleus, which is a relay center for signals to the cerebellum. The reticular formation, which is a loose network of nuclei extending through the entire brain stem, containing things like the cardiac center, the vasomotor center, and the respiratory centers. Pons above that would develop from the metencephalon. It's about 2.5 centimeters long, about an inch. Its broad anterior bulge in the rostral is rostral to the medulla. It posteriorly consists of thick stalks called cerebral pendicles. The cerebral peduncles, as I should have said before, are stalks on the posterior pons that connect it to the cerebellum. Here we have cranial nerves 5, 6, 7, and 8, which have sensory roles in hearing, equilibrium, taste, facial sensation, as well as motor roles in eye movement, facial expression, chewing, swallowing, urination, and secretion of saliva and tears. The reticular formation is also in the pons. It has additional nuclei, and here they are generally concerned with sleep, respiration, and posture. From there, we move up to the midbrain, which it develops from embryonic mesencephalon. It's a short segment of the brainstem that connects the hindbrain to the forebrain. Some of the anatomical features here are the cerebral aqueduct, the continuation of the medial lemniscus and the reticular formation, and there are also motor nuclei of 2 cranial nerves that control eye movements, cranial nerve 3 and 4, the oculomotor and trochlea. We have the tectum, which is the roof-like part of the midbrain posterior to the cerebral aqueduct. It has 4 bulges, the superior colliculi, which is involved in visual attention, tracking moving objects, and some reflexes. 2 inferior colliculi, which relay signals from the inner ear to the thalamus and other parts of the brain. The cerebral peduncles are the 2 anterior midbrain stalks that anchor the cerebrum to the brainstem. The tegmentum is within the cerebral pundicle, dominated by the red nucleus. It has a pink color due to high density of blood vessels. Its connections go to and from the cerebellum and are involved in motor control. The substantia nigra is a nucleus within a peduncle. It's dark nucleus pigmented with melanin. It's a motor center that relays inhibitory signals to the thalamus and the basal nuclei, suppressing unwanted body movements. And it's problems here in the substantia nigra that lead to the tremors of Parkinson's disease. The cerebral crus is a bundle of nerve fibers that connect the cerebrum to the pons and carry corticospinal tracts. That reticular formation that we've been mentioning is a loose web of gray matter that runs vertically through all levels of the brainstem and even up into the upper spinal cord. Occupies the space between the white fiber tracts and the brain nuclei, has connections with many areas in the cerebrum, and it has more than 100 small nuclei that do not have these distinct boundaries. You can see it has projections and connections throughout the brain. That is involved in somatic motor control. It's involved in cardiovascular control, pain modulation. It's very important in sleep and consciousness, and habituation, the activity in the cerebral cortex that ignores repetitive, inconsequential stimuli. The cerebellum is the largest part of the hindbrain, second largest part of the brain as a whole, contains more than half of all brain neurons. We have granule cells found in the cerebrum, Purkinje cells from large cerebellum neurons. Anatomically, it has cerebral hemispheres connected by a bridge known as the vermis. The superficial cortex of gray matter has folds or folia, which branching white matter making up what is literally known as the tree of life, the arbor vitae, and the deep nuclei. It gets inputs to the from the cortex and gets input to the cerebellum, goes to the cortex, output comes through the deep nuclei. So it comes from the external downward. The cerebral peduncles are 3 pairs of stalks that connect the brainstem and the cerebellum, and their fibers carry signals to and from the cerebellum. Inferior connects the medulla oblongata, most of the spinal input. The middle connects to the pons, and the superior connects to the midbrain. And here is that tree of life that is called, the arbor vitae, in white matter, and then the gray matter, taking the planned movements to a large extent and fine-tuning them so that they can be carried out smoothly. So that motor coordination and locomotor ability. It's got sensory, linguistic, emotional, and other non-motor functions that are becoming more and more evident. It's very important for comparing textures of objects, perceiving space, recognizing objects from different views, keeping judge of elapsed time, and maintaining a tapping rhythm. Directs eye movements to compensate for head movements. Helps in verbal association tasks, planning, scheduling, and even emotional control. Cerebellum is a very important part of the brain. The forebrain has the diencephalon and the telencephalon. Diencephalon has three major components: the thalamus, the hypothalamus, the epithalamus. That thalamus is a mass on each side of the brain. It's perched at the superior end of the brainstem beneath the cerebral hemisphere. Constitutes about 4/5 of the diencephalon and is composed of at least 23 nuclei with 5 major groups: anterior, posterior, medial, lateral, and ventral. The thalamus is the gateway to the cerebral cortex, so almost all input to the cerebrum synapses in the thalamic nuclei. It processes information on the way to the cerebral cortex. Not all that information gets passed along. The thalamus is also screening out most of the information, doesn't feel necessary to move up. Plays a key role in motor control, relaying signals from the cerebellum to the cerebrum. Provides feedback loops between the cerebral cortex and the basal nuclei. We know it's involved in memory and emotion. It's part of the limbic system. The hypothalamus forms part of the walls of the floors of the third ventricle, extends anteriorly towards the optic chiasma, posteriorly toward the mammillary bodies. Those mammillary nuclei relay signals from the limbic system to the thalamus. Attaches to the pituitary through a stalk-like structure called the infundulum, and contains many nuclei with a wide variety of visceral, emotional, and behavioral functions. It is a major control center of the autonomic nervous system as well as the endocrine system. It's involved in the homeostatic regulation of most body systems. We'll be talking more about the autonomic nervous system later this week. It controls things like hormone secretion, autonomic effects, and thermoregulation. This is where the thermostat is for your body, the hypothalamic thermostat, monitoring body temperature. Has structures that regulate food and water intake, sleep and circadian rhythms. It's involved in memory and emotional behavior and sexual responses. The epithalamus is a small mass where we find the pineal gland, which is part of the endocrine system. We'll check that out in the second semester. The habenula is a relay for the limbic system to the midbrain, and there it forms a thin roof covering the third ventricle. The cerebrum develops from the telencephalon as the largest, most conspicuous part of the human brain. This is our seat of sensory perception, memory, thought, judgment, and voluntary motor actions. This is our entire universe. Everything you know about the world, you know because of your cerebrum. Gross anatomy, it has 2 cerebral hemispheres divided by a longitudinal cerebral fissure. It's connected by a white fibrous tract called the corpus callosum. The gyri and the sulci increase the amount of cortex in the cranial cavity. This is the folding that I was referring to when we started. Allows for more information processing capability. Each hemisphere has 5 distinct lobes, and 4 of them are named for the cranial bones that cover them. The frontal lobe is in front of the central sulcus. It has a region that involved in voluntary motor functions, motivation, foresight, planning, memory, motion, mood, social judgment, and aggression. The parietal lobes are between the central sulcus and the parietal occipital sulcus. They are integration centers for things like general senses, taste, and some visual information. In the back, we have the occipital lobe, which is the primary visual center of the brain. The temporal lobe has a lateral and horizontal. Below the lateral sulcus, it functions in hearing, smell, learning, memory, some aspects of vision and emotion. And then the one that is not named for a bone is the insula. It's deep to the lateral sulcus, and it's important in understanding spoken language. Gives us information about whether we like something in as far as taste goes, or also whether we're being cheated. Gets information from visceral receptors. Most of the volume of the cerebrum is white matter. This would be glia and myelinated nerve fibers that transmit signals. They form tracts, which are bundles of nerve fibers in the central nervous system. Those tracts can be projection tracts, commissural tracts, or association tracts. Projection tracts extend vertically between higher and lower brain and spinal cord centers. Commissural tracts come from one cerebral hemisphere to the other, allowing for communication from both sides of the cerebrum. The association tracts connect to different regions within the same cerebral hemisphere. Long association fibers connect with different lobes, short ones connect within the gyri of a lobe. The gray matter layer, which is about 2 to 3 millimeters thick, is the covering surface of the hemisphere. It makes about 40% of the brain mass, has between 14 and 16 billion neurons. There are also stellate cells and pyramidal cells. 90% of the human cerebral cortex is neocortex. It's the 6-layered tissue that has a recent evolutionary origin. The limbic system is an important center for emotion and learning. It's got the prominent components present in each hemisphere include the cingulate gyrus, the hippocampus, and the amygdala. These components are connected through a loop of fibrous tracts that allow somewhat circular patterns of feedback. They also connect to the frontal lobe and give us our emotional responses, which we can hopefully modify with our frontal lobe for conscious awareness. The basal nuclei are masses of cerebral gray matter buried deep in the white matter, lateral to the thalamus. They're involved in motor control. Include the caudate nucleus, the pitumen, and the globus pallidus. All three are collectively called the corpus striatum because they have a striped appearance, and the lentiform nucleus is the pitumen and the globus pallidus. And they are shown here. See them on this side. That integration that happens in the brain, we are getting more and more information, slowly learning about it, but we don't have all the answers yet. But we do know that the sleep, memory, cognition, emotion, sensation, motor control, and language are controlled by integration. That involves the interaction between the cerebral cortex, the basal nuclei, the brainstem, and the cerebellum. They don't have easily defined anatomical boundaries. It's a combined action of multiple brain levels. We can use an electroencephalogram to record brain waves, which are rhythmic voltage changes in the surface layers of the cortex. These can give us information about studying normal brain functions at sleep versus consciousness, help in the diagnosis of degenerative brain diseases, including those with metabolic abnormalities, brain tumors, or trauma. Lack of brain waves is the common criteria for what we would refer to as brain death. There are 4 types of brain waves which differ in amplitude and frequency. Alpha waves are between 8 and 13 hertz. We see these when we are awake and resting with our eyes closed and our mind wandering. They're suppressed when the eyes are open or when you are performing a mental task. We do not generally see them during sleep. Beta waves have a frequency of 14 to 30 hertz. They are accentuated during mental activity and sensory stimulation. Theta waves are between 4 and 7 hertz. We see those in drowsy or sleeping adults. Delta waves are less than 3.5 hertz. They have a very high amplitude, however, and we see those in adults in a deep sleep. That sleep is a temporary reversible loss of consciousness from which we can be awakened when stimulated. It is one of the body's circadian rhythms, events that reoccur at intervals of about 24 hours. They occur in distinct stages, which can be recognized with an EEG. In stage 1, you feel drowsy, your closed eyes begin to relax, and we go from alpha waves of mixed frequency. Stage 2 is a light sleep. Brain waves have a lower frequency but higher voltage, and see sleep spindles, which are high spikes. Stage 3, we get moderate to deep sleep. The theta and delta waves begin to appear. Vital signs drop. In stage 4, we get a deep sleep. Muscles are relaxed. Vital signs are at their lowest level. And then we periodically backtrack and enter a stage of REM sleep. So that rapid eye movement or REM sleep occurs about 5 times a night. The sleep backtracks from stage 3 or 4 to stage 2. The eyes oscillate, move back and forth. It's often also called paradoxical sleep because the EEG resembles a waking state. Sleeper is harder to be aroused than during any other stage of sleep, and this is where we get vivid and long dreams. Sleep paralysis is stronger. That prevents dreams from being acted out. And the parasympathetic activation is going to cause penile or clitoral erection and constriction of pupils. The rhythm of sleep is controlled by interactions between the cerebral cortex, the thalamus, the hypothalamus, and the reticular formation. The suprachiasmatic nucleus in the hypothalamic nucleus that serves as a master biological clock. It gets input from the eye. It controls things like sleep, body temperature, urine production, hormone secretion. Has output pathways to the dorsomedial nucleus and the pineal gland, and that pineal gland secretes melatonin and acts on the reticular activity system to block stimulatory signals from the thalamus to the cortex. Waking is initiated by orexins, which are neuropeptides secreted by the hypothalamus. They stimulate the reticular activating system, which then allows the thalamus to resume sending stimulatory signals to the cortex. Awareness of the environmental stimuli leads to waking up. In the condition known as narcolepsy, orexin levels are low, and that leads to excessive daytime sleeping. Sleep does have a restorative effect, and sleep deprivation can be fatal in experimental animals. Sleep may be the time to replenish energy sources such as glycogen and ATP. REM sleep may consolidate and strengthen memories by reinforcing some synapses while eliminating others. Cognition is a range of mental processes by which we acquire and use knowledge. Sensory perception, thought, reasoning, judgment, memory, imagination, and intuition. It's accomplished by widely distributed association areas of the cerebral cortex. And those association areas constitute about 75%, 2/3, 3/4 of the brain. We learn about cognition from studies of patients with brain lesions, so people who have problems with cognition. But we are getting more and more information from studies in what we would call normal individuals using both PET and fMRI. Examples of cognitive function in the associated cortex. So the parietal lobes help perceive and attend to stimuli. So if an individual has a lesion, they can cause contralateral neglect syndrome. They can be unaware of objects on the opposite side of the body. Temporal lobe helps us to identify stimuli. Individuals with lesions in the temporal lobe can get agnosia, the inability to recognize, identify familiar objects, prospagnosia, can't recognize faces. Frontal lobe helps us think about the world and plan and execute appropriate behaviors. People with lesions in the frontal lobe can get personality disorders or have inappropriate social behaviors. Memory is information management by the brain. It's going to involve learning, which is acquiring new information, memory, which is storage and retrieval of that information, as well as forgetting, eliminating the trivial things and remembering the important ones. Amnesia is a defect in explicit memory, inability to describe past events, and it can be anterograde or retrograde. Anterograde, you're unable to store new information, whereas retrograde amnesia, the person cannot recall things that happened before the injury. The hippocampus is an important region for memory. It's part of the limbic system. It functions in memory consolidation, teaching the cerebral cortex until a long-term memory is established in the cortex, organizes cognitive information into a unified long-term memory, but does not hold the memory itself. The cerebellum is involved in learning motor skills. The amygdala is important for emotional memory. Emotions and feelings involve interactions between the prefrontal cortex and the diencephalon. That prefrontal cortex, or the frontal association area, is a seat of judgment, intent, and control over expression of emotions. Feelings, like fear, arise from deeper brain regions, the hypothalamus and the amygdala. The amygdala is especially important in fear, but it also affects food intake, sexual behavior, drawing attention to novel stimuli. It gets input from sensory systems. It has outputs to the hypothalamus, which influence somatic and visceral motor systems. Your heart can race, your blood pressure rises, hair stands on end, vomiting. The prefrontal cortex is important in controlling the expression of those emotions, the ability to express love, control anger, and overcome fear. Behavior is going to be shaped by learned associations between the stimuli, our responses to them, and whether there's reward or punishment that results. Sensation happens in the primary sensory cortex. There are sites where sensory input is first received and one becomes conscious of the stimulus. Those have association areas near the primary sensory areas that process and interpret the sensory information. And they are multimodal. The associations receive input from multiple senses and integrate it into our overall perception of the surroundings. Special senses include vision, hearing, equilibrium, taste, and smell. So vision is in a primary visual cortex, which is the far posterior region of the occipital lobe. It gets visual stimuli. Visual association areas border the primary cortex anteriorly and occupy all the remaining occipital lobe. Hearing, we have a primary auditory cortex in the superior region of the temporal lobe that receives auditory signals, and an auditory association areas in the temporal lobe that are inferior to the primary cortex. They are used to recognize spoken words, music, and voices. Equilibrium, the signals from the inner ear project to the cerebellum and brainstem nuclei. Taste and smell, we have a primary gustatory cortex getting taste signals, and the primary olfactory cortex getting smell signals. The general senses, the somatosensory, somatic, and somatic, are distributed all over the body. They involve relatively simple receptors. That's really the difference between special senses and general senses. They include senses of touch, pressure, stretch, movement, heat, cold, and pain. The signals from the head pass through the cranial nerves to the brain. Signals from the rest of the body ascend sensory tracts of the spinal cord. The thalamus processes the input from the contralateral side. There are selective relays to signal the postcentral gyrus or the parietal lobe. They have somatosensory association areas that make cognitive sense of the stimulus. The sensory homunculus is a diagram of sensory inputs primary to the somatosensory cortex of the parietal lobe. Sort of resembles an upside down caricature of the contralateral side of the body, with the size of the different regions of the body being determined by the number of inputs that come from there. That somatotopy is a point-to-point correspondence between a region of the body and an area within the central nervous system. So the receptors in lower limbs project to the superior and medial parts of the gyrus. And that's how we know where the information is coming from. Motor control, we have motor association, or the premotor area located where the intent to contract the muscle begins, found in the frontal lobe. And then it's programmed and transmitted to neurons through the precentral gyrus, the primary motor area, and they go through the brain stem and spinal cord, as well as the cerebellum, to fine-tune those controls. There is also homunculus here. Neurons for toe movement are deep in the longitudinal fissure on the medial side, and at the summit we have the trunk, shoulders, and arms. Again, the size of the body parts are represented by the number of motor outputs that you have, not the size of the body. Here's an example of that homunculus. Upper motor neurons are pyramidal cells of the precentral gyrus. Their fibers project caudally. There's about 19 million fibers ending in the nuclei of the brainstem. Most of the fibers desiccate into the lower medulla and oblongata, and from the lateral cortical spinal tracts on each side of the spinal cord. In the brainstem or spinal cord, the fibers of the upper motor neurons synapse with the lower motor neurons and innervate skeletal muscles. Basal nuclei and cerebellum are also important in motor control, muscle control. Motor functions of the basal nuclei would include the onset and cessation of movements, repetitive hip and shoulder movements of walking, highly practiced learned behaviors. Dyskinesias are movement disorders caused by lesions in the basal nuclei, which lead to abnormal movements. Parkinson's disease is a good example of that. Other brain regions involved in motor control, the cerebellum, aids in learning motor skills, maintains muscle tone and posture, smooths the muscle contractions, coordinates eye and body movements, coordinates motion with different joints with each other. Here we get lesions would lead to ataxia, which is a clumsy, awkward gait. There are different brain regions responsible for components of language: reading, writing, speaking, and understanding words. The posterior speech area, traditionally known as Wernicke's area, usually in the left hemisphere, is involved in recognition of spoken and written language. Motor language area, the Broca's area, is the inferior prefrontal cortex, again usually in the left hemisphere. It generates the motor program for muscles of the larynx, tongue, cheek, and lips, allow for speaking. When we intend to speak, Wernicke's area formulates the phrases and transmits the plan of speech to Broca's area. Broca's area transmits the program of primary motor cortex commands to the lower motor neurons, supply the relevant muscles. We also have emotional aspects of language controlled by regions in the opposite hemisphere. An aphasia is a language deficit from lesions in either Wernicke's or Broca's area. We can have non-fluent aphasia or fluent aphasias. Cerebral lateralization is the difference in structures and functions between the two cerebral hemispheres. I want to start out right now by telling you that you are not left-brained or right-brained. No one is, no matter what you've heard. Neither hemisphere is dominant. Each is specialized for certain tasks, but everyone uses both hemispheres about equally. The left hemisphere is the categorical hemisphere, usually specialized for spoken and written language, has sequential and analytical reasoning, and it breaks information into fragments and analyzes it. The right hemisphere is usually the representational hemisphere. It perceives information in a more integrated way. It's a seat of imagination and insight, more musical and artistic skills, perception of patterns and spatial relationships, comparisons of sights, sounds, smells, and taste. Lateralization is correlated with handedness, age, and sex. Left hemisphere is categorical hemisphere, 96% of right-handed people and 70% of left-handed people. Lateralization develops with age. Children are more resilient to lesions on one side, and males exhibit more lateralization than females and suffer more functional loss when only one hemisphere is damaged. The cranial nerves are 12 pairs of nerves arising from the base of the brain. They exit the cranium through foramina, lead to muscles and sense organs located primarily in the head and neck, with one big exception. Most of the fibers of the cranial nerves begin at nuclei of the brainstem and lead to glands or muscles. The motor fibers. Sensory fibers begin in the receptors located mainly in the head and neck and lead mainly to the brainstem. Most cranial nerves carry fibers between the brainstem and the ipsilateral receptors and effectors, and lesions to the brainstem cause sensory or motor defects on the same side. There are exceptions. The optic nerve, half of the fibers desiccate, and the trochlear nerve, all the afferent fibers lead to a muscle of the contralateral eye. So the nerves, the sensory nerves, C1, 2, and 8, carry signals only from outlying sensory organs to the brain. The motor nerves, C3, 4, 6, 11, and 12, stimulate muscles but also contain fibers of proprioception. And the mixed nerves, 5, 7, 9, and 10, have sensory functions that might be quite unrelated to their motor function. Facial nerve, 7, has a sensory role in taste and motor role in facial expression. The olfactory nerve, 1, is purely sense for smell, has separate fascicles that pass through the cribriform plate to the olfactory bulb. The optic nerve, 2, is purely sensory. It's a nerve for vision. It carries visual signals from the retina to the brain. Ocular motor, 3, is a motor nerve controlling 4 of the 6 muscles that are involved in eye movements. Trochlear nerve, number 4, is a motor nerve controlling 1 of the 6 muscles of eye movement. The trigeminal nerve is the largest cranial nerve, most important sensory nerve of the face. It's divided into 3 branches. 5.1 is the ophthalmic, 5.2 is the maxillary division, and 5.3 is the mandibular division. 2 sensory and 1 mixed. The abducens, number 6, is a motor nerve controlling 1 of the 6 muscles of eye movement. The facial nerve is a mixed nerve, has motor functions for facial muscles, sensory functions to taste of the anterior 2/3 of the tongue. It has 5 branches: the temporal, zygomatic, buccal, mandibular, and cervical. The vestibulo-ocular nerve, number 8, is a predominantly sensory nerve of hearing and equilibrium, has a motor effect on the inner ear, tune the sense of hearing. The glossopharyngeal, number 9, has sensory functions. It's sensation from the tongue, throat, upper ear, regulation of blood pressure and respiration, but it also has motor functions in salivation, swallowing, and gagging. The vagus nerve, number 10, is the most extensively distributed of any of the cranial nerves. This is the one that does leave the head and neck region. Sensory functions include taste sensations of hunger, fullness, gastrointestinal discomfort. Its motor functions are involved in swallowing, speech, acceleration of the heart, bronchoconstriction, gastrointestinal secretion, and motility. The accessory nerve, 11, is a motor nerve involved in swallowing and head, neck, and shoulder movements. And number 12 is the hypoglossal nerve. It's a motor nerve involved in tongue movements, speech, food manipulation, and swallowing. There are some cranial nerve disorders. Trigeminal neuralgia is a recurring episode of intense stabbing pain in trigeminal nerve area near the mouth or nose. Pain can be triggered by touch, drinking, washing the face even, and that may require cutting the nerve. Bell's palsy is an inflammation around the facial nerve, leads to weakness of facial muscles, a sagging mouth on one side, or an eyelid. The mind is why we really like to study the brain, and this is where integration goes to where we don't really have a good idea, but we are getting more and more information. This is going to involve in studying with positron emission tomography to visualize where blood flow increases when certain regions of the brain are being active. We can also use functional magnetic resonance imaging to look at an increased blood flow to an area. Magnetic properties of hemoglobin depend on how much oxygen is bound to it. So both of these tell us when we are thinking of something or imagining something, which regions of the brain are becoming more active. And that leads us to the end of the brain and the spinal cords. I will be posting this with the homework, and then this week we will also do